My name is Adriana Arneson. I'm a bioinformatics scientist in the Target Enrichment R&D team at Twist Bioscience. And in this presentation, I'm going to give you an overview of how we use Twist custom panels to do high throughput and cost-effective sequencing-based genotyping. So first, a little bit of background. As you might be familiar, genotyping arrays have been the workhorse of the genetics community for characterizing genetic variation in a high throughput and genome-wide fashion. Now, in past years, as the cost of next-generation short-read sequencing has gone down exponentially, some researchers have started to shift towards sequencing their DNA in order to call genotypes. And actually, one of Twist's most popular products are our human exome kits, which allow our customers to enrich their DNA samples for the exome using our bioattenuated probes, so all their sequencing is targeted in this region of interest. And the important thing about hybrid capture panels is that our probes are actually quite tolerant to mismatches to the sequence they're trying to capture, especially when the mismatches are in a continuous part of the probe, which is what is shown in this first figure and is also about what you can expect from SNPs and short indels. So as you can see, even with 4 mismatches to the target region, we still get approximately 80% of the capture efficiency of a probe with no mismatches. So knowing this, we wanted to show how our custom panels, in which we can design these probes to target any region we want in the genome, can actually be used to get very cost-effective genotyping calls that come with all the advantages of sequencing, which I will get to in a bit. So to show this, we designed a genotyping panel that targets approximately 200,000 SNPs in intergenic regions, which were characterized by the Genome in a Bottle Consortium, so that we can actually judge the accuracy of our genotyping calls against a gold standard. But first, in terms of the actual capture, we show here our Picard metrics at a raw sequencing depth of 150x coverage. We get excellent metrics which are very comparable with our exome panels, both in terms of mean observed coverage and percent target based at a certain coverage. As you can see, this tracks closely with our exome. We also see very few zero coverage targets. We see a small duplication rate, low dropouts, and a very acceptable amount of off-target, especially because, bear in mind, these are intergenic regions, much more GC poor than coding, so capture here can actually be a little bit tricky. So, after convincing ourselves that the capture performance is reasonable, we evaluated how well our variant calls actually perform in comparison to genotyping calls using a genotyping array. So in this figure, and I apologize if it's a little bit hard to see this right side, but the values in here are actually represented in this figure as well. We see that we have very high SNP precision and recall across 3 samples, which all come from different ancestries. And most importantly, because we have sequencing data here, we can actually call indels as well with over 90% precision and sensitivity. So, and then the absolutely most important part here is that these metrics actually hold in much lower coverage as well. So here we're talking about around 50x mean coverage. Even at 20x mean coverage, we see over 99% SNP precision and sensitivity. And even when we go down to 10x mean coverage, which amounts to only about 0.84 gigabytes of sequencing, we still see very high precision and a sensitivity that is well within the realm of what could be used to impute the variants that we do miss at this lower coverage. And so now, one thing that can affect hybrid capture assays, which people sometimes are worried about, is that the DNA containing the reference allele against which you actually design your probes might be easier to capture than the one containing the alternate allele. And although we've addressed this, when we looked at our tolerance to mismatches, we wanted to more explicitly reassure ourselves that this doesn't cause any problems. So this can be termed as allele reference allele bias. So what we looked at here, in the NA12878 samples, we looked at the percentage of reads supporting the alternate allele across all our calls. So if there's no bias whatsoever, then this would follow a binomial distribution with mean 0.5, because each read would essentially come from either allele with probability exactly 0.5, which is the gray line in each of these panels, since you're effectively flipping a coin, since you expect to see both alleles with the same frequency. So what we actually observe in the panels is this green line across all our SNPs, and then stratified by the GC content of the SNPs in these other of the region in these other panels. So the bias is actually quite small. It tracks quite well with the expectation, and although there is a little bit of it, the difference here between the green and the gray line is noticeable. It is actually on par with what literature shows can just happen from short read aligners, which actually have a preference to align reads that match the reference genome. And as we saw previously, our variant calls are still highly accurate. And so now finally, one of the most important benefits of our panel and of our technology is that because we have this highly respected exome, we can actually just spike in the SNPs to our exome panel, and we can tweak the concentration so that most of your sequencing is still going towards the exome, but you still get capture in the SNPs. So for example, in this figure, what I'm showing you are targets from our exome and replicates, the coverage in replicates from exome captures. And these are SNP targets. And as you can see here, we mix the exome and the SNP in a 2-to-1 ratio. So now the sequencing coverage you see, you get it across the exome targets, and you also get it across the SNP targets, just about at around 50% of the coverage in the exome. So this saves you a lot of hassle because you can actually do both exome and genome sequencing and genotyping in the same with the same library prep and in the same hybridization experiment. You just need to sequence a little bit more. And again, it might be hard to see here in this panel, but what I'm showing you is that our capture metrics in this kind of blended panel between the exome and the SNPs are actually quite good, and they track with what we see in the exome quite well. So that's it. Thank you for listening to this presentation.